Hello students, welcome to CBSE NCERT Solutions by Vedantu. I am Ms. Gopika, your own biology master teacher. So yesterday we did molecular basis of inheritance part 1, where we understood what is the introduction, or what is DNA, what is RNA, how are the bonds formed, and why are these bonds formed, how are these bonds formed. All these questions we answered, and then we also understood two experiments which proved that DNA is the genetic material and the biochemical nature of DNA, right? Today we are going to continue from there. We are going to continue with the next experiment called as Hershey and Chase experiment. Now ma'am, why was that previous experiment rejected and all of that question? I have answered you in the last class that biochemical nature that was done by Avery McLeod McCarthy experiment did not become a success because of the reason that DNA, the experiment did not give them a clarity on is DNA the genetic material, because yes, you use DNAs, protease and RNAs, and the results were also there, but then again two results were there. So all this brought a confusion. Okay, that is the reason why Avery, McLeod and McCarthy's experiment did not become a success. Then the most accepted, widely accepted, see, remember students, you do an experiment. Okay, you are like, okay, water is this or water is neutral. You do an experiment. Now that experiment, you proved that water is neutral and its pH is somewhere around 7. Now just because you proved it, it is not going to be widely accepted. Okay, always remember this. Just because you proved it, it's not going to be widely accepted. It has to be accepted by a set of zoologists, botanists, chemists, and other scientist people who are working on the same experiments, who has studied about these. So same thing has to happen here also, that okay, I am telling that DNA is the genetic material because I proved that it is the genetic material. It does not work like that. So like that, widely accepted, when I'm telling, understand that it is accepted by all the people around, or all the zoologists and botanists who were working on this experiment. They were the one who understood that yes, according to Hershey and Chase and according to their experiment, what they have proved is absolutely right. So let's see what the experiment was. So students, Hershey and Chase took a bacteriophage. Now for those students who doesn't know what is bacteriophage, it is a virus that infects bacteria. Students, I'm really disappointed that a lot of you are watching the lectures, but none of you are liking the sessions or marking your attendance or commenting about how you feel about the session, which is very sad, because you know that we are doing this completely for your benefit. Okay, and all you have to do is just just give that appreciation or that compliment possible, which will motivate or boost the teachers, right? So that you guys are not doing. I'm very disappointed in that. So Hershey and Chase took a bacteriophage, that is a virus that infects bacteria. So let's try drawing the bacteriophage. Okay, this is how the bacteriophage is going to look like. Yesterday we drew the same structure for your pyramid, if all of you remember. Okay, this is the structure, and inside this genetic material is going to be there. So ma'am, can you label this and tell us what you have drawn? So students, what you see outside is a protein coat. So I think all of you know that virus, when we talk about it, is made up of two components: a protein coat. Why this protein coat is there? To make sure that the genetic material, be it RNA, be it single stranded RNA, DNA, to protect this genetic material. Okay, this protein coat is there, and then the genetic material is there. Protein coat is there and the genetic material is going to be there. So virus is made up of this. So here you can see this is the protein coat and this is the genetic material. This is the genetic material. Now this, there is tail fibers. Okay, this is like your collar. Okay, this is like your collar, and this is going to be your tail fibers. Now what they are going to do, this bacteriophage, when they are going to infect a cell. Okay, let's take this. Okay, this is the E. coli cell. I'm just drawing one cell. Okay, any of the bacteria. Let's say this is the E. coli cell. Okay, E. coli cell. what this bacteriophage is going to do is, students, it is going to attack the cell, and using its tail fiber, it is going to hold on to this bacteria, and it is going to release its genetic material inside the bacteria. Okay, it's going to release its genetic material inside the bacteria, and this we call it as infection. Now, why is a debate going on that is virus living, is virus non-living, is viruses who? That is because of the main fact that only if a host is there, this virus is activated. Yes or no? If a host is not there, this virus will not do anything, because the virus becomes dangerous when it meets the host and when it puts its genetic material inside the host. Agreed or disagreed? So the question about is virus living, is virus non-living, comes from the fact that this virus is dangerous because it meets the host and it infects the host by releasing the genetic material into the cell. Understood, all of you? So that is the main reason. So this is a bacteriophage. Okay, this is a bacteriophage. So this is the experiment that they are trying to do. So what they did, students, they took two radioactive substance, okay, one S35 radioactivity and one P32 radioactivity. Now, ma'am, what is this S35 P32? Why was the reason that they took this? Is simple. S35 or sulfur is only present in protein. Okay, only present in protein. Agreed? Only present in protein. And P32 is only present in DNA. Okay, only present in DNA. Now, when I'm trying to strain something, when I'm trying to show that, okay, this is the final result, I have to radioactively label it so I can see it. Okay, I can see it. So what they did was, when they dipped or when they, let's say grown the bacteriophage in S35 medium, they noticed that the bacteriophage protein code, the protein code got highlighted. Okay, let's draw with this color. The protein code got labeled, radioactively labeled. Got it? So S35 we are denoting with this color. Okay, and when they notice that, okay, they notice that P32 we are going to denote with orange color. P32 okay, P32 students, they noticed when they cultivated, they saw, when they did this, they noticed that the DNA got radioactively labeled. Now what can you expect? You can expect that, okay ma'am, in the last, we can notice that if it is DNA, we are going to see it, okay, we are going to see P32. That means radioactivity is going to be inside the cell. Okay, radioactivity is going to be inside the cell. See, if it was protein, very simple, students, if infection has to happen, if it is protein that is going to act as the transforming principle, then the protein code should be inside the cell, right? If it is DNA, DNA is already inside the cell, it is going to make an infection, it is going to make a change. So this is the experiment. Okay, now we will go to the diagram given in NCERT. So here you can see, okay, here you can see bacteriophage, okay, bacteriophage, radioactively S35 labeled protein capsule. So obviously this is going to be labeled as S35 because protein, sulfur is only present in protein. Okay, here you can notice that the red, students, the red indicate radioactivity. Okay, so radioactively labeled P32. So infection happened. Okay, infection happened with both, one with S35 and one with P32, where you can see one the protein code, one the genetic material. Okay, after the infection happens, students, what do you notice? So here you can see infection has happened. You will notice that the genetic material has got inside. Here also genetic material got inside, but was it radioactively labeled? It was not radioactively labeled. Okay, so here what happened? First step is going to be infection. That is where the bacteriophage, okay, bacteriophage is going to infect the cells. Okay, infect the cells. Got it? Second is going to be, see, because this protein code is going to be present on the top of the E. coli. Now imagine something is present like this. What will I do? I'll shake it. No, I'll shake it so it falls. Or what I'll do? I'll do blending or mixer. With a mixer, I do blending so that this protein code goes outside. So blending is using a mixer. Okay, using a mixer blender, you blend it, or you just give a rotation so that the protein coat gets removed. Protein coat which is attached to the outside of the cell, no, protein coats get removed. Now here, if you notice, you will notice that there was no radioactivity in the cells. So if protein was the genetic material, then we should have noticed radioactivity in the cell, no? But here you will notice that no radioactivity in the cell. You will see all the radioactivity in the supernatant. Now you will be like, ma'am, what is the centrifugation? Students, centrifugation is a technique where we use the centrifuge tubes. Okay, these are the centrifuge tubes. In this, we'll add our sample, and at a speed, okay, RPM we tell. So at some RPM we will do high rotation. Okay, rotation will be at greater speed. Now what is going to happen? We are going to get two things, right? We are going to get two things. We are going to get a liquid or a lighter portion on the top. Okay, we are going to get a lighter portion on the top, and we are going to get the heavier particle. We are going to get the heavier particle at the bottom of the centrifuge tube. Yes or no? If I rotate something, or if I mix mud and water and I let it for settling, what will happen? The sand will come down, the water will go up. So same thing we do here, centrifugation will happen. Okay. Now whatever I got, the lighter portion, the top portion is called as the supernatant. What is this called? Supernatant. And the down solid portion which I'm going to get, which is heavier, is going to be the pillet. So genetic material, we expect it to be heavier. It will have to be seen in the pillet. Okay. So radioactivity detected in cells, that is, here you will notice that the cells are able to see the radioactivity, or in the cells we are able to see the radioactivity, and we will see no radioactivity detected in the supernatant. Why? Simple thing, because DNA will be present in the pillet. DNA is a heavier particle. It's a genetic material. It got into the cell. It infected the cell. So this experiment was a complete change. Okay, this experiment actually stood away from all other experiments, proving that DNA is the genetic material. So Hershey and Chase experiment proved that DNA is the genetic material. So if the question comes, who proved DNA is the genetic material, your answer shouldn't be Griffith, your answer should be Hershey and Chase. Because understand that Hershey and Chase was the one who proved that DNA was the genetic material. If the question comes, who was the first person to identify that something is getting transformed from one cell to another cell, your answer should be Griffith. Otherwise, your answer should be Hershey and Chase. So now that you understood the concept, you try to read NCERT. This way you will be able to understand. 3 steps are there: infection, blending, and centrifugation. Infection is where the bacteriophage will affect the bacteria, okay, and release their genetic material inside. Second will be blending, where we remove the protein coat which is attached to the outside of the cell. And the third step is centrifugation, where we rotate the whole tubes at a higher RPM, at a higher speed, and the radioactive genetic material will be inside the cell, which is going to affect the cell and will be seen in the pillet of the cell and not in the supernatant. Got it? Not in the supernatant. Now students, like when we stand for election, imagine you want to stand as the MLA, you want to stand as something. When you stand for election, there will be some criterias, no? Some criterias. The criteria should be that you are 18 plus to vote. No, not for election, even for voting also. You should be an Indian citizen. Okay, you should be an Indian citizen, etc., etc., etc. Things will be there. Now, these are the criteria. Same way, if DNA or any RNA, DNA, protein, whoever wants to be the genetic material, they have to go through some criteria telling that I am actually capable of being a genetic material, because being a genetic material is not a joke. Why? Because they have to do inheritance. Yes or no? They have to do inheritance. They have to make sure that all the information from the parent has to come to the offspring without any leak of information. At the same time, they should be able to participate in central dogma. They should be able to convert DNA to mRNA to protein. All this they have to do. Transcription, translation, everything has to be to the point. So let's see what are the things that will give the criteria, or that has made DNA the genetic material. It should be able to generate its replica. So what have they told? It should be able to generate its replica, which is called as replication. That means it should be able to make its own copies. Make its own copies. Now I have given you, imagine I have given you a Xerox paper, okay, a main copy, and I've told you that you should take the Xerox and return it back to me. Now imagine you haven't taken the Xerox, or you have not made a copy for yourself. Obviously in the next class you're going to get scoldings from me asking, I had given you the Xerox copy, why didn't you make the copy? Same way DNA also, students, has to make its own copy, okay, using it itself as a template. It should be able to form the dotted strands. Now it should be stable chemically and structurally. That means what? Chemically they should have a strong structure, that they should not be highly reactive, okay, they shouldn't be unnecessarily or highly reactive. If our DNA is highly reactive, you can just imagine the consequences, because it is going to react for the smallest inconvenience caused in our body, smallest environmental change, it is going to start reacting, smallest change in the temperature, in the weather, everything would have changed the genetic material. So it should be stable chemically and structurally. Structurally, I have already mentioned this to you guys, that because of the absence of a OH, absence of an O, this DNA has become more stable and chemically stronger than your RNA. Now, it should provide the scope for slow changes. Now, you do not want to sleep as a human and wake up as a penguin. Do you want to? Okay, you want to wake up as a penguin? You cannot. So, slow mutation should happen. It should happen. It should take years to make a small modification, to make a small change. We are not like cockroaches, we are not like mosquitoes, that okay, today I put DDT on this batch of cockroach, next cockroach generation is going to be resistant to DDT. We cannot, we humans are not like that, and for us slow mutation should happen. Okay, slow mutation should happen, slow changes should happen. Okay, now resistance is just an example that I gave, but you should understand that it is more than just resistance. Okay, now it should be able to express itself in the form of Mendelian characters. That is, they should be able to express itself from the Mendel's character, from principles of inheritance you already learned. No, be it any character or trait, it has to express that, and it should have the ability to express the Mendelian characters. Got it? These are the four main criteria to being the genetic material. It cannot be just randomly telling that today I'll be the genetic material. One day carbohydrates wake up and carbohydrates is like, you know what, I'm actually bored of being this energy source, I just want to be the genetic material. They cannot be the genetic material. Understood? So students, see, the first point about replication is absolutely right, because our DNA replicates. Okay, replication happens in a different method by itself, okay, which you're going to study next. Second thing is about the stability. Now remember that one OH, okay, which was not present, okay, which was not present in the second carbon, okay, which gave an H, this was more than enough to make it less reactive. Okay, less reactive, more stable, okay, more stable. And also one more thing is, students, they are less reactive, more stable. And the next fact is also that the presence of I don't know how many of you have thought of it, presence of thymine instead of uracil. Okay, thymine instead of uracil. This is also a factor, yes or no, don't you think? Because uracil is present in RNA, but presence of thymine. Okay, at the same time you have DNA, students. At the same time you have RNA. RNA has a OH here, and because of this OH, RNA is highly reactive, very reactive. Okay, very reactive, and it is less stable. Okay, less stable. And then what? It has uracil instead of thymine, which makes it less stable. And not only that, students, this fellow does mutation. Okay, this fellow, I'm telling, like, he's my classmate. Okay, RNA does mutation. Okay, it does mutation, but fast mutation. See, first of all, RNA is highly reactive. So you can just imagine, if it is so reactive in the body, what is going to be the after effects of it, right? What is going to be the after effect of it? It is going to make all the problems possible, because it's very reactive. It's like going there, it's doing a reaction, going here, going here, doing a reaction. You know, there'll be someone in your home, no, who's very short tempered. Wherever they go, they'll find a problem to make, or they'll make a problem, or they'll shout. Yes or no? They'll shout, they'll make a problem, they'll make all noise everywhere. So like that, if you imagine, like in our body there's one fellow who will go and react with everyone. We do not want that. And at the same time, fast mutation. Here it is going to be slow mutation. Okay, slow mutation. So this makes RNA the genetic material. So students, RNA has fought war to become the genetic material. Okay, it's not like RNA got it easy. You will not get anything in your life easy. Everything comes with the pain, gain, sacrifice. Many things happen. Okay, so DNA, RNA. So RNA understood why it is not the genetic material. See, I will not tell it is not a genetic material for anything. It is the genetic material for virus, but RNA also has other jobs. Okay, RNA is going to do other jobs. What are the other jobs RNA is going to do? It's going to act like a catalyst, messenger, adapter, okay, and much more. So all this job also it is going to do. It is not just going to be like, okay, I'm not the genetic material, I will not help you guys, you do whatever you want, I will not help you. That attitude RNA doesn't have. Okay, in body everything is in love with each other, everything is in harmony. Only humans outside there is a problem. Okay, inside the body everything is in harmony with each other, everything tries their best level, you know, to actually uplift each other and all of that. Got it? Yes, uplift each other and all of that. So if one examines each requirement one by one, because of the rule of base pairing and complementary, both the nucleic acid DNA and RNA have the ability to direct their duplication. The other molecules in the living system such as proteins fail to fulfill this criteria. Like the question can come, no ma'am, why didn't you not even consider protein? Protein is very important. The problem is protein does not even have any features. Okay, now the genetic, to become the genetic material, the genetic material should be stable enough not to change with different stages of life cycle, be it age or with the changes in the physiology of the organism. Understood? Just because you have gone from teen to adult, do you expect your DNA to change? No. So stability as one of the properties of genetic material was very evident in Griffith's transforming principle itself, that the heat which killed the bacteria at least did not destroy some properties of genetic material. It's not all the properties, because if all the properties were there, then the mice would have died, okay, during the heat kill estate. But there were some properties that remained that allowed, you know, some properties that remained that made the R strain virulent. Yes or no? Some properties remained. Some properties remained. Now, this now can be easily, see, which killed the bacteria, at least did not destroy some properties of the genetic material. This now can easily be explained in the light of the DNA, that two strands being complementary, if separated by heating, come together when appropriate conditions are provided. Students, see, if two people are there who really like each other, or you know, they feel like, okay, you are the one for me, I'm the one for you, then see, during a heated argument they will go separated, right? They will separate themselves, saying it is too much of heat. That is same thing that is going to happen in DNA. We are giving the heat, the DNA strands are going to get separated. But as the heat goes down and as the condition gets favorable, we expect that these people come back together. This is what true love is. Okay, so DNA has true love. So DNA has true love, and this is how we also should be, that when that heat of the moment comes, we should actually go away, so that we do not tell anything rude to our parents, especially because I always notice that, you know, at this age, especially at your age where you are a teen, you know, even I have been there, but you know, that age from 17, or let's say from 13 onwards, okay, 13 to around 20, 21, even 21, I'll say, you have a lot of heat with your parents. Okay, the heat is so much that you want to just, be a volcano that erupts for no reason. Okay, that is because you feel like, okay, you start seeing the generational difference. You start seeing the gap that your parents are not being cool, but you're very cool. Your students at college are very cool, or maybe their parents are cool. So like there are 101 requirements, problems just keep popping up during this age. You might think that, ma'am, after 18 I might be very mature. So let me tell you, 18 is just a maturity for you to go and vote. Other than that, you do not get any maturity, because your hormones are still in an imbalanced condition. It is still trying to fluctuate as much as possible. So whenever that fluctuation is going to happen, you're going to have that feeling that actually outside people are better than my family is so boring. My parents are not cool enough, they're very old, and 100 things will come to you, and you will start seeking attention from outside the world. And that is when things go bad, and that is where I have taught you in reproductive health as well. Drug abuse happens, smoking happens, because all that looks cool. Okay, and that is when your parents will come into the picture telling that, don't have this bad habits, do this, why are you weak in studies? Because automatically you become weak in studies. Okay, and then right now we have a strong social media where everyone is accessible to free internet. Everyone is access not I don't mean free internet, but definitely we all have wi-fi's at home where we have access to internet 24/7, right? These things did not exist. So what is going to happen is, you should always remember that when that heat is going to happen, you know that you're going to tell something so rude that you might hurt your parents forever. Just go away. Just tell them that, please give me the time, I will come back to you and I'll talk to you, but just right now, just give me that break. Okay, I know they might not be practical at that moment, because they feel like, okay, why is he doing this, or why is she doing this? But after the heat is over, when you go back to them and talk to them calmly, let me tell you, the damage that you could have done by telling few words, or by telling that, you guys are the worst parents, I should have got a better parents, or my friends' parents are very cool, you guys are like this. Please do understand that they have their own problems. They have been grown up with a very bad background. It could be the generational trauma and the inheritance that they have carried from their parents also. So that's the reason why they were not able to be cool, because they didn't no one facilitated them. But once you become a doctor, once you become an engineer, once you do a job which pays well, which does well for you, then you are going to provide them. And that is automatically when you see your parents to be cool, and you will notice that, oh my god, my mom was so cool, my dad was so cool, I never knew about it. It's because you will also reach a maturity, you will also reach a realization stage. They will also reach a stage where they can remove the burden from themselves and be like, okay, my son and daughter is taking care of everything, I can just relax. When they relax, automatically that pressure of being a parent, being a good parent, automatically comes down. Okay, so this is from my life experience. I know no one wants to listen to lectures, no one wants to listen to motivation, but it's not a motivation. Students, do not take it as motivation. I do not like giving motivation. I have always told this. I'm not a teacher who likes to give a lot of unnecessary motivations, and because I believe that motivation is just like sugar coating something. Like, okay, I want to paint this bottle, I put a paint, but inside whatever is the color, it's going to be the color. So motivation should come from within. But I'm going to tell you, what I just told is a fact that you will experience up to the age of 21, and after 21 you will actually change your perspective about life. You will realize that earning every rupee is very hard, and having your own money is a big responsibility. Spending your own money is a responsibility, and money can change your life. Job can change your life. Like, all these factors are going to revolve around your life. Okay, so till that you will be like, oh, you cannot give me for this, you cannot do me this, all this you will tell now, but later you see it will change. Okay, so you should also be like this, like DNA, that when heat is there, they're going to get separated. Okay, and when the heat goes down, or when the condition becomes favorable, they're going to come back together. Now, for the 2OH group present at every nucleotide in RNA is a reactive group and makes RNA liable and easily degradable. That is also there, that easily RNA will get degraded. Okay, if the genetic material get degraded, students, you can only imagine, no, that when we find, because of some accidents, because of some burning, half of the body is burned or 90% of the body is burned, we have still a hope that we'll be able to identify this person with the help of DNA. Right, but imagine RNA was the genetic material. If a person's body gets burned, or if there is any condition, environmental condition, we cannot find, or we cannot do the genetics test. Okay, we cannot do the test. Clear? All of you? . Sure, all of you are clear. Yes. So that's the thing. So we need a genetic material that will be able to withstand any kind of temperature. Now see RNA, okay. RNA is also known to be catalytic, hence reactive. Therefore, DNA chemically is less reactive and structurally more stable when compared to RNA. Therefore, among the two nucleic acids, the RNA is a better genetic material. Okay, RNA is a better genetic material. Whatever I told, same thing is here. In fact, see, the presence of thymine at the place of uracil also confers the additional stability of DNA. Now, detailed discussion we will understand later. But look here, both DNA and RNA are able to mutate. In fact, RNA being unstable mutate at a faster rate, which we don't want. Okay, virus having RNA genome and having shorter lifespan mutate and evolve faster. Yes, students. Now from past 1 week I had rhinovirus allergy, that is I had common cold plus I had dust allergy. Okay, common cold, I'm going to write cold common because that is what is in my life. Okay, cold is so common, common cold. So rhinovirus, every time it affects my body, every single time. This is my body's immune system, this is my body's immune system, this is my body's immune system. Why my immune system is not able to recognize it? Maybe it recognized at the first instant, it's not able to recognize at the second, maybe at the third instant it's not able to recognize. Why? Because these viruses come in variants. You know, they come in variants. Now, what do I mean by variants, students? Every time different varieties of rhinovirus come, that my body is every time realizing that, oh my god, new pathogen has come, new foreign particle has come. But it is not new, it is the old fellow who has come in different, you know, like that Kamala Hassan movie, 10 roles he is doing, you know, like that. This rhinovirus is doing different roles every day in my body, because of which my virus is not able to get protected, and the medicines which I'm eating also is not able to differentiate, oh, today who came, like, you know, every day you like one person pops up. So like that, this comes in variants. And why these variants are coming? Because of the fact that they can mutate faster and they can evolve faster. Okay, mutate faster and they can evolve faster. Understood? Got it? Now see, RNA can directly code for synthesis of protein, hence can easily express the characters. DNA, however, is dependent on RNA for synthesis of protein. The protein synthesis machinery has evolved around RNA. So here when we talk about Mendelian characters also, what do we expect them to do? We expect them to get converted from DNA to RNA, RNA to protein. So here they're trying to say that if RNA was the genetic material, there is only one step, okay, one step process. Okay, but because since DNA is the genetic material, we have a two-step process. Why am I telling two-step process? Because from DNA to mRNA, mRNA to protein, two steps. Okay, first step, second step. But if RNA is the genetic material, only one step. Okay, only one step. Understood? I've explained every line of NCERT. Now it's your job to study the chapter thoroughly. Okay, study the chapter thoroughly. Now, from the foregoing discussion, an immediate question becomes evident: which is the first genetic material? It shall be discussed in detail in chapter of chemical evolution, but briefly we shall highlight some facts and points. Okay, RNA is the first genetic material. So here, students, the biggest reveal has happened. Okay, the biggest reveal of the time, that RNA is the genetic material. RNA is the genetic material. RNA is the first genetic material. This word is important. Okay, from RNA, DNA got evolved. Because obviously this is a single stranded structure. Okay, single stranded structure. This is the double stranded structure. So this evolved from this. Okay, there is now enough evidence that suggests that essential life processes such as metabolism, translation, splicing evolved around RNA. RNA used to act as a genetic material as well as a catalyst and important biochemical reaction in living system that are catalyzed by RNA catalyst and not by protein enzymes. But RNA, being a catalyst, was reactive and hence unstable. Therefore, DNA has evolved. So definitely, see, when you go from one phase of life to another phase of life, you actually become a better person. Now, why am I telling this? Because here RNA was the first genetic material. When it got evolved to DNA, or from RNA DNA was evolved, DNA became a better person. Okay, it became a better person. Why? Because it got chemically, structurally stable compared to RNA. So DNA has evolved. See, DNA has evolved from RNA with chemical modifications that made them more stable. This I already told you, made more stable, and they became complementary to each other. They formed double strands, and they also had nitrogenous bases to keep them together. So students, here you should remember that this your RNA acts as a catalyst. I already mentioned, no? It also acts like a messenger RNA. It acts like an adapter RNA. Okay, adapter RNA. Okay, it acts as an adapter RNA. RNA. First genetic material. Okay, RNA is the first genetic material, and we have to remember that RNA, from RNA evolved DNA. Okay, from RNA evolved DNA, and this DNA is more stable chemically and structurally. Okay, chemically and structurally it is more stable. Now we should also remember that RNA can act as catalyst. Okay, messenger, adapter. Okay, they can act as all of this, and all of this character, students, is very, very, very important. Okay, it's important for the central dogma. Okay, important for the central dogma. Yes or no? See, this fellow adapter, that is your tRNA, has to be the one who is going to read the translation, that is going to read the code on the mRNA. Then we have messenger, that is your mRNA, which is going to code for protein. Then catalytic job also it does. So this is very, very important, right? We cannot take RNA as like, oh, you are not the genetic material, so you are just, useless. No, it is not useless. It is forming, or it is helping in most of the body activities. Yes or no? It's going to help in most of the molecular biology activities. Now, replication. Now, what is replication? See, first point in your criteria was that DNA, or whoever is going to be the genetic material, has to replicate. Okay, has to replicate. See, RNA also will replicate. Okay, it's not like RNA won't replicate. It has to replicate. Now, DNA replicates in a very different way. And who found this? It was found by Watson and Crick in the year 1953. Okay, in the year 1953, they found that there was a copying mechanism for genetic material. That is where they form, with the help of a template, they'll be able to form a new DNA strand. Now, I'll explain you this with an example that you can see in your home. I always use this example. So, see, students, this is, I think we have to draw it a little big. Okay, thick. What is this? Don't tell me, ma'am, it's a braid. Okay, it's not a braid, it's your genetic material. So this is parent. This is parent DNA. Obviously, if I'm going to tell you what is replication, make a copy. I have given you a Xerox copy. Okay, let's say I have given you just a original copy. I don't have any books here. So original copy of something. 1 second, students. I have given you original copy of something. I told you go take Xerox and come. Now I have given you 2 copies, 2 sheets. Okay, because DNA is complementary. 2 sheets are, 2 pieces are going to be there. So I will, simple thing, we will take one strand. We'll take one strand. Okay, which is the template one new thing. Okay, we will call, students, remember, the paper that I give you to take Xerox is going to be the template. The one that you're going to get the Xerox is going to be the Xerox copy. So using this one template, okay, we are going to form the daughter DNA. Okay, we are going to form the daughter DNA. Okay, we're going to form the daughter DNA. One second, students. I'm getting a call from office. Okay, yes, this is going to be your daughter DNA. So this is the template, the actual copy I gave you. This is the Xerox that I got. Okay, this is the Xerox that I got. Understood? Now, students, both of them, both the parents, this is also one parent. Okay, this is template 2. Okay, and this is the daughter, D2. Yeah, okay, this is going to be daughter 2, D2. Okay, now students, normally what happens? Okay, I give you the Xerox copy, you came back to me, you are going to give back the Xerox copy. So I'm going to get a parent DNA. Okay, I'm going to get a parent DNA, parent DNA, and I'm going to get a daughter DNA. Okay, daughter DNA. Okay, daughter DNA. Got it? Okay, so here parent DNA, here daughter DNA. This is normally how a normal replication happens. Now I call this replication, not I, okay, people have called this replication. This is called as conservative replication. Conservative replication. Conservative replication means what? Parent DNA was there, daughter DNA was there. Parent DNA acted as a template and formed the daughter DNA. Second, complementary. So obviously they're complementary strands. So strand 1, strand 2 form the daughter DNA. After the replication was done, both the parents became together, both the daughters became together. But our DNA is thoda special, students. Wait, I have to use this. Our DNA is very, very special. I'll tell you why. So in your home, imagine this is your parent. Okay, this is the parent. This is your parents. Okay, parent 1, that is your father. Okay, let's say mother and father. Okay, mother, let's put father and mother, because always we tell like that, no, father and mother. Now they are going to so let's say they have come to solve your issues. Okay, one, let's say father is with the elder daughter. Okay, father is with the elder daughter. So father will act as a template, students. Okay, remember father means your parent DNA, parent 1, okay, father, and the daughter DNA. Okay, daughter DNA. Got it? Father and the daughter DNA. Then the next, okay, next is going to be mother and the next daughter DNA. Okay, daughter 2, D2. This is replication, what I taught you just now. Template is there, daughter is going to be born, template is a daughter. Now what is going to happen in your home, I'm going to tell you. Okay, imagine you and your sister fought, or you and your brother fought. Father is going to take elder daughter's side, mother is going to take younger daughter's side, or vice versa. Now after the fight, when they come to solve the fight, what will happen? They will, like, both of the parents, okay, both of the parents are the daughters are there, they're like trying to solve the fight, but in middle, father and mother will get into a fight. They'll tell that, you taught her like this, you became like this, like when blame game they'll start playing, and suddenly father and mother will start fighting. It was initially your fight, but what happened in the middle, father and mother started fighting. Now if father and mother starts fighting, do you think they'll go back together? They will not go back together. So they will make, what they're going to do, they are still going to remain a gang where the father and the daughter will become one gang. Then again another daughter, another mother, and another daughter will become another gang. That means, students, in our DNA, what is going to happen? It is going to be one parent DNA, one daughter DNA, one parent DNA, one daughter DNA. So this is called as semi-conservative. Semi-conservative. I hope all of you understood this. If you understood this, definitely don't forget to comment. So students, conservative means what? Till here it was conservative. That is, this fellow acted as a template. Okay, first strand of DNA acted as a template. We formed a new daughter DNA. Okay, we formed a new daughter DNA, or let's say a new strand. Okay, which is going to be same thing happens here. But unfortunately, what happened, because the fight went so bad, parents also fought. They became a gang, that is parent and the daughter, parent and the daughter. So this is called a semi-conservative type of replication, and our DNA does semi-conservative type of replication. There are there are conservative structures, there are semi-conservative, there is dispersive. This is the three types of replication that happens. But our DNA have choose a semi-conservative discontinuous technique. Okay, that is where the parent and daughter will become together. So now that was proven with an experiment. Which was the experiment? The experiment was Meselson and Stahl experiment. Meselson and Stahl experiment. Okay. Now what is this experiment, ma'am? Experiment was that they grew E. coli in a medium containing NH4Cl, okay, N15, where N15 is the heavy isotope of nitrogen. Okay, where N15 is a heavy isotope of nitrogen as the only nitrogen source for many generations. The result was that N15 was incorporated into newly synthesized DNA. So first they grew it in N15. So N15 you consider as the parent. Okay, parent. Then this heavy DNA molecule could be distinguished from the normal DNA by centrifugation in cesium chloride CSCl density gradient. Please note that N15 is not a radioactive isotope. It can be separated from N14 based on density. One is heavy, one is light. Now then they transferred the cell into a medium with normal N14 and took the samples at different intervals. I'll teach you with the help of a diagram. See students, this is the N15. So this is a culture plate. Okay, this is the culture plate. Okay, this culture plate. Okay, this culture plate is N15 medium. Okay, so we grew the E. coli bacteria. Got it? We grew the E. coli bacteria. Now, once we grew the E. coli bacteria, we notice that both the strands have become N15. N15, N15. After that, we grew the E. coli in another medium. Okay, we grew the E. coli in another medium. Okay, which has N14. Okay, N14. Okay, N14. One was heavy, students. This is heavy. This is light. Then we notice that one strand was N14. Another strand was N15. One strand was N14. One strand was N15. That means what? One was parent. One was daughter. Okay, these two were parents. So this continued, okay, and why did they choose E. coli? They chose E. coli because E. coli's replication period is around 20 minutes. Okay, E. coli's replication period is around 20 minutes. Yes or no? So E. coli's replication was around for 20 minutes. So 20 minutes, if they are able to replicate, that is the easiest thing to do, yes or no? Right? If otherwise you have to wait for 48 hours for some replications. Okay, you cannot wait for 48 hours, then the whole experiment is going to get waste. So they chose E. coli because of the replication time of 20 minutes. Students, duplication is around 18 minutes, replication is around 20 minutes. So you have to know that. Okay, some students will have doubt, ma'am, why is in this book I saw it's 18 minutes. That's because of the duplication. Replication is different. Okay, slightly different. So you can that's the time difference. Now see here, N15, this becomes the parent. Okay, this parent, after the 20 minutes you kept it for replication, you notice that in generation 1 you got hybrid. Hybrid means what? Parent, daughter, parent, daughter. So you got parent, daughter, N15, N14. Then after 40 minutes when you saw, you got N14, two hybrids. Okay, so you got what? You got 50% hybrid, you got 50% light. Obviously you will get light. Why? Because simple thing, I have dipped, let's say, a white cloth in blue color stain. After that I lifted from there and I put it into yellow color for a longer time. Then I got blue and yellow. I got like that, what was that called, students? Oh no, I forgot. During that COVID time it was very famous. Tie-dye t-shirt. Okay, so it became tie-dye, yellow plus blue it became. Then again I kept it for 40 minutes in yellow color. Okay, again I kept it in 40 minutes for yellow color. So what is going to happen? Blue stain is going to slowly fade because yellow is going to take it up. Okay, same way parent DNA was N15, and after some time obviously it's going to fade as and when it moves, no? So here what you have to learn is, see, does the DNA that was extracted from the culture one generation after the transfer, okay, of N15 to N14 medium, that is after 20 minutes, E. coli divides in 20 minutes, had a hybrid or an intermediate density, okay, hybrid was formed. DNA extracted from the culture after another generation, that is after 40 minutes, was composed of equal amounts of hybrid and DNA, like DNA, that is 50-50. So if E. coli was allowed to grow for 80 minutes, then what would be the proportion of light and hybrid density DNA? This is a question to you, okay? Now, very similar experiment involving use of radioactive thymidine to detect the distribution of newly synthesized DNA in chromosome was performed by your Faba, okay, Faba beans, by Taylor and his colleagues in 1978. Okay, this experiment proved that DNA in chromosomes also replicate in semi-conservative manner. Now, students, we'll answer that question. The question is, first I keep it in N15 medium. Okay, I am going to get 2 strands. I love to teach with a lot of colors because students tell me that, ma'am, I actually remembered your notes. I actually remembered the portion where you wrote it with green, pink. Even I was a student like this. I used to make my own notes and I used to always remember that because of the colors sometimes, you know. I used to be like, oh, in the book side, I wrote something with pink color. Now, what does that? I used to remember like that. So first you kept it in N15 medium, okay. After you kept it in N15 medium, you transferred it to N14 medium. Okay, you transferred it to N14 medium and you notice that one strand was N14. Okay, another strand was, I should have drawn it this way, students. Okay, another strand was N15. Then again after 40 minutes you keep it. You notice that you got, I'll draw like this, okay. You got 2 hybrids and you got 2 lights. Okay, again you keep it for 60 minutes. What are you going to get? See, here you got 50% hybrid and 50% light. Yes or no? Here what you're going to get, students, you are going to get, you know how, I'll tell you how to do this. See, you separate all the yellow strands. 1, 2, 3, 4. Okay, separate the blue strands also. 1, 2. So now you are going to get each strand is going to make a copy. Yes or no? So here what you are going to get? You are going to get 75% hybrid. Sorry, 75% light. So first I'll write hybrid. Wait, 25% hybrid and 75% light. Got it? 25% hybrid and 75% light. Got it? Light. Now next one, okay, they wanted to find after 80 minutes. So you go 80 minutes. I think 80 minutes, I don't have place to write it here. Where do I write it? Yeah. Okay, so now after this 80 minutes, okay, 80 minutes. Now 60 minutes done. Now students, how many strands did you get? You got 6 strands. Okay, 1, 2, you have to keep separating them. Okay, 3, 4. Okay, this ones, then you had 4 of them. Okay, so 1, 2, 3, 4. Got it? So 6 to 8 we formed. Okay, 6 to 8 we formed. So 1, 2, 3, 4, 5, 6, 7, 8. Okay, this many strands you got. Now this is what, this is going to be around, we can say approximate, okay, 12.5% of hybrid and around you can just do, how much is this? This is around 1 to 6, no? In 1 second, I'll just count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 I had to draw. 12 I have to draw. So here 1 2 3 4 5 9 10 11 12. Okay, then only I'll be able to do the calculation. Otherwise like this also it's enough, but good no calculation. 1 2 3 4 5 6 7 8 9 10 11 12 12 no. So 10 around so here students you can write that how many strands of this will be light DNA. You will get around 87.5% of light strands, okay, light DNA. So what I basically did is I separated each of the strand. Okay, 1 2 3 4 5 6 7 8 9 10 11 12 12 strands I separated. 12 strands I separated and I started adding them. So what I got after 80 minutes, I noticed, okay, after 80 minutes I'm almost going to get 12.5% of hybrid and 87.5%, where you will see 12 12 total strands are there, out of which 10 is your light and 2 is your hybrid. Okay, 10:2. Understood? That ratio you will get. Then again you can do for 100 and you can keep doing it. Okay, you can keep doing it. So this is what it is. So you basically separate the strands, add the strands and you check. Okay, but the values are going to be this only, students. It is going to be very simple. Like we write it otherwise also. Okay, we write it without even doing all this, because this is a long process. No, we do not get that much time when you are writing an exam. So you remember this 20 minutes. Okay, 20 minutes, first 20 minutes you are going to get you're going to get 100% hybrid. Okay, 40 minutes you're going to get 50% hybrid and you're going to get 50% light. Okay, then in 60 minutes you're going to get 75% light and I'll write it in order. You're going to get 25% hybrid and 75% light. Okay, then in 80 minutes you're going to get 12.5% hybrid and around 87.5%, again this is approximate, okay, light. So this is how the calculation goes. Okay, by 80%, 100% it will go more or less. So this is how the calculations. 20 minutes you will get full hybrid, 40 minutes you will get half, 60 minutes you will get 75-25, 80 minutes you will get 12.5 to 8.7. So this, if you remember this box, if you remember, it's more than enough. Okay, more than enough. This box is very, very important. So this, if you remember, students, very important. Okay, 20 minutes. It won't work. Okay, never mind. Okay, is this clear? So this is the actual hybrid. So this is how replication happened. So now, students, it just got stuck. Yes. So whenever we are doing this one, now if you go back to this, you will be able to understand. Okay, that first we kept it in N15, that is the parent. After 20 minutes we got hybrid. After 40 minutes we got more 50% of light, 50% of hybrid. Okay, and remember that they are going to do centrifugation, and centrifugation is going to happen with your cesium, okay, chloride, which is going to help in the centrifugation. Okay, centrifugation for separation of the strands. Remember, and why did we choose E. coli? Because of the replication time of 20 minutes, which is easier to replicate. Okay, which is easier to replicate, and you will call the parent daughter as hybrid. Okay, you will call this as hybrid strands. Hybrid strands, and this whole thing is called as semi-conservative. This whole thing is because your DNA is semi-conservative. Semi-conservative means half conservative. Okay, half conservative. Yes, students, before we go to machinery, let's take a break. Okay. I'll see you guys soon. Hello students, I'm back. Okay, so I hope all of you had your food as well. Okay, because what happens is between classes, sometimes we forget about our health. So we should all think about our health. Health is everything. Especially as a biology teacher, I totally believe that health is everything, and there's nothing more than health. So always take your enough breaks. It's okay. We can definitely catch up even after 10 minutes. Nowhere, I'm not going anywhere. You're not going anywhere. Okay, so breaks are good. We should refresh ourselves, and especially for teachers, when we're taking multiple classes, everything pains, throat, eyes, everything. Okay, so that's why. Okay, in living cells, so students, now we understood what is the kind of replication that is going to happen. That is going to be semi-conservative replication. That is where the parent and the daughter forms a hybrid. Instead of going back and forming the parent DNA, daughter DNA, what they're forming one parent, one daughter, one parent, one daughter. So hybrid is formed. But ma'am, how did this strand get separated? Our DNA strand is complementary to each other. How did this strand get separated? How did they form the new strand? All these doubts you're going to have. So that is when NCERT introduces you to a topic called as enzyme machinery, where you talk about the different enzymes. Okay, you talk about the different enzymes and you understand how the semi-conservative replication happens. So let's draw a DNA, students. I'm going to explain you in the best way possible. All you have to do is just trust the process and keep learning. Okay, keep learning. See, this is a DNA. Okay, and we start unzipping from here. Unzipping means what? Both the strands of DNA is like this. I slowly start unzipping it. Now it has become, obviously students, I cannot twist my hand and break, okay, break my bones. But then just imagine this is the replication fork, okay, and this is the twisted strands, okay. This is my strands. Okay, this, imagine this is the two strands. Oh my god, yeah, this is the, I think I'll just remove the adapter. Yes, this is the two strands. Okay, this is the two strands. They have wound around each other. Okay, they are wound around each other. Okay, this is how they are wound around each other. Okay, now what I'm going to do, because they're wound around each other, I have to make sure that I unzip them. So the structure is going to exactly look like this wire. Okay, here you're going to see a replication fork like this Y-shaped structure, and this unzipping you're going to see. Okay, so slowly this will start unzipping like this, unzipping like this, unzipping like this, and they will form a two strands. Okay, I cannot cut this, students, obviously, charger. Chalo. So unzipping. So this is the strand. Okay, I try to draw as much as better possible with the pen. Okay, yeah, this is the strand. Here the unzipping has started. Okay, here the unzipping has started. Okay, unzipping has started. Now this unzipping happens, why? Because of an enzyme. Okay, we have an unzipping enzyme called as helicase. Okay, what do we have? An unzipping enzyme called helicase, which will unzip. Got it? Which will unzip. Is it clear to all of you? Yes. Okay. Chalo. So unzipping has happened. That is because of an helicase enzyme. So the goal is that, students, the twisted DNA, we have to make sure that slowly we are unzipping and two strands are coming out of it, right? So the unzipping enzyme is going to be helicase. Now, ma'am, after they unzip, we have to start forming new strands. So how is it going to be? So students, once I unzip this people, okay, once I unzip this people, this is going to be the next stage. Okay, I'm just going to draw it this way because it's easy for us to explain. Okay, let's go 5 strand to 3 strand and 3 strand to 5 strand. Okay, once we unzip, this is how the structure is going to be. Okay, ma'am, unzipping happened. Again, here the DNA is going to be there. 2.2 meters long DNA means, again, the unzipping is going to continue. Now, to form a new strand, we are going to introduce the hero called as DNA polymerase. Okay, DNA polymerase. What is DNA polymerase? Students, I forgot my glasses and I cannot stay without glasses now because of my migraines. So let me just call and tell. Just 1 minute. Okay. No, it's a glass അപ്പുറത്തെ റൂമിലുണ്ടെന്ന് തരുമോ കണ്ണാടി? Yes, okay. So DNA polymerase, he is the hero. I'll tell you why he is the hero, because he is going to form the new template. He is going to form the new template, means new daughter DNAs. Okay, he is the one who is going to form the new daughter DNAs. Okay, got it? Now, problem is that DNA polymerase is that hero who cannot understand where to start, what to start, when to start. Okay, where to start, what to start, when to start. So since he cannot understand where to start, what to do and what not to do, he is going to take the help of one person called as the primase. Okay, he's going to take the help of who? A primase. This primer, okay, this primer is going to add, okay, in the front so that the DNA strands can form. But this hero, no students, hero will tell that no, set is not okay. Okay. Set is not okay, that is not okay, this is not okay, the heroine I don't like. So like that only, this DNA polymerase has two conditions. First condition is, I will only work on a strand if the strand is single. Okay, he'll only work on a strand if the strand is single, and he will only work on a strand which is from 5' to 3'. He will only work on a strand which is from 5' to 3'. Understood? Now, this is the two condition. Okay, people were like, okay, you are the hero, fine, we will just agree to you. Now the problem is, this fellow doesn't know where to start. He doesn't know until the director tell action, he doesn't know he has to start. So the primer will come and add. Okay, the primer will come and add, and the DNA polymerase will start forming the new strand. DNA polymerase will start forming the new strand without any problem from 3' to 5' direction. Okay, 3' to 5' direction. Okay, that problem got sorted. Now there is one more problem. The second strand that we have is from 3' to 5'. What did this fellow tell? I will only work on a strand whose polarity is from 5' to 3'. Then everyone got tensed. What we will do? We have to finish the movie. We have to get this done. So then primer was like, don't worry, I will come to help. I will do one thing. Anyway, DNA polymerase is little blind. Okay, he you know, primer is like, I can totally handle it. This fellow is thoda blind. So what I will do, I will start adding the primer from behind, because where the primer adds, that's where the DNA polymerase will start forming the template. So the DNA polymerase will start forming template. Because it is going in reverse direction, student, more places the primer has to keep adding. Okay, because it is going reverse. Okay, reverse, more places the primer had to keep adding, so that this DNA does not understand that it is doing the work in reverse pattern. Okay, in reverse pattern, like this. Now what is going to happen? The primer got added. Okay, we formed a strand which is from 5' to 3'. Okay, we formed a strand from 5' to 3'. Now almost problem is done. Now the strand which was formed without any issues means from the primer continued, it kept continuing. This is called a continuous strand. This is called a continuous strand. Okay, or leading strand. Okay, or leading strand. The strand that made the problem, telling that no, I will not be able to form, this strand will be called as the discontinuous strand. Okay, discontinuous strand, or the lagging strand, or the lagging strand. Now students, there's one more thing. Each of these fragments, okay, each of these fragments which you're seeing here is called as Okazaki fragments. Okazaki fragments comes from a scientist name. Okay, each fragments are called Okazaki fragments. I hope my handwriting is clear and visible. Yeah, no? Okay. It's called as Okazaki fragments. Now this DNA polymerase showed its real color. DNA polymerase is like, I don't want this primer and all, it is blocking my way. So let's do one thing, let's remove the places wherever primer was there. He's like, let's remove the places wherever primer was there. I will form the new nucleotide, that is, I will continue the new nucleotides here. Okay, I'll continue this. But students, since there are there are pieces, fragments, to keep these fragments together, to bind these fragments together, we are using a glue. We are going to use a glue to stick these people together. Okay, to stick these people together, and this glue that is going to act like a glue is called as ligase. Okay, ligase. You will study this glue again in your biotechnology chapter. This is the glue that is going to stick the fragments. Okay, or glue the fragments. Stick the fragments. So which are the enzymes actually there for all of this? Who is the enzyme? Helicase, DNA polymerase, ligase, and primer. Got it? These are the 4 enzymes that are going to help in replication. Is this clear to all of you? Any doubts? Any doubts? Clear? These are the 4 enzymes that is present in your machinery of your entire replication. Okay, that is, students, very simple. See, this strand was formed smoothly. 3' to 5' was a continuous leading strand, no problem. Here, from 3' to 5' we formed the 5' to 3'. No, this is called as discontinuous strand or the lagging strand, because they are in fragments, and those fragments are called Okazaki fragments. To stick all these fragments together, we have ligase or a glue which will stick the fragments. Got it? Then we have helicase, who is going to unzip the DNA fragments. That is, they are going to remove this complementary. These people are all like this, no? They will slowly unzip and come. I hope it is clear to all of you. I hope it is clear. Okay. Now this Y-like structure which you're seeing, okay, this Y-like structure which you're seeing is called, okay, is called as the replication fork. Replication fork. This, what you're seeing, is called the replication fork. Now we will go back to NCERT. Okay, see, in living cells such as E. coli, the process of replication requires a set of catalyst or an enzyme. The main enzyme is referred to as DNA dependent DNA polymerase. DNA dependent means it is dependent on the DNA, because the template is going to be DNA. If template is DNA, then only DNA polymerase will work. Since it uses a DNA template to catalyze the polymerization of deoxynucleotides, that is the formation of new nucleotides. These enzymes are highly efficient enzymes, as they have to catalyze polymerization of a large number of nucleotides in a very short time. Okay, E. coli has only 4.6 into 10 to the power 6 base pairs. Compare it with human, whose diploid content is 6.6 into 10 to the power 9 base pair. Completes the process of replication within 18 minutes. Okay, so here they have given replication 18 minutes, there they have given 20 minutes. It's actually duplication and replication, so don't get that confused. Okay, that means the average rate of the process of replication within 18 minutes, that means the average rate of polymerization has to be approximately 2000 base pair per second. Not only do these polymerases have to be fast, but they also have to catalyze the reaction at high degree of accuracy. Yes or no? If they do not form the strand properly, students, entire machinery will fail. Yes or no? Entire machinery will fail. So everything has to be accurate. Now, any mistake during the replication would result in mutation. Okay, even one thing changes, one nucleotide changes, it can cause you a syndrome. Okay, it can cause you a genetic syndrome. Yes or no? Okay, now furthermore, energetically, replication is very expensive process. Deoxynucleotide triphosphates serve dual purpose. That is, in addition to acting as substrates, they provide energy for polymerization reaction. That is, the 2 terminal phosphates in deoxynucleoside triphosphates are high energy phosphates. So students, here they're trying to say that this replication will utilize energy, and the energy that we utilize is going to be your ATP. Okay, ATP, and that is deoxynucleoside triphosphates. Okay, triphosphates. So there'll be sugar, nitrogenous base, then there is going to be phosphate group, one more phosphate group, one more phosphate group. Breaking of these phosphate groups are going to yield a lot of energy. It's an energy consuming process. Understood? Now, in addition to DNA-dependent DNA polymerase, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in its entire length due to a very high energy requirement, the replication occurs within a small opening of DNA helix referred to as replication fork. I told you, the Y-shaped structure is called as a replication fork. The DNA-dependent DNA polymerase catalyzes polymerization only in one direction, that is 5' to 3' direction. This creates some additional complications at the replication fork. Consequently, on one strand, the template with polarity 3' to 5'. So here look student, the DNA-dependent DNA polymerase catalyzes polymerization in only one direction, that is I told you 5' to 3'. And the strand has to be single, that means the strand has to be unbound. Okay, so the strand that DNA polymerase is going to form here, that is 3' to 5' is going to be continuous. Okay, whatever I explained only, okay, and 5' to 3' is going to be discontinuous. The discontinuous will be synthesized by DNA ligase, that means they will be joined by DNA ligase. Yes or no? Clear, all of you? Now, the DNA polymerase on their, I forgot to tell you one thing. Okay, here we'll go through that. The DNA polymerase on their own cannot initiate the process of replication. Also, replication does not initiate randomly at any place. There is a definite region in the E. coli DNA where the replication originates. Such regions are called ORI. So, students, remember, even like when we teach you plasmid and all of that, you know, ORI is called as origin of replication. This is the place where the replication is going to start. Replication is going to start. So ORI is very necessary so that replication happens. Okay, and some places you can also read that ORI is a place where the adenine and thymine is there because AT bond is easy to break. Okay, easy to break, but this is again additional information. Researchers have been happening about more about it. You do not have to worry about it. Okay, now very important questions can come from here for your boards. Because of the requirement of the origin of replication, there is a piece of DNA if needed to propagate during the recombinant DNA procedure. This is what you will study in biotechnology. Recombinant DNA technology. That is, if I want to add a foreign particle, I have to make sure that the the vector that I am taking should have an ORI site, okay, or a plasmid. Okay. Now the vectors provide the origin of replication. See, vectors means vehicles. You study in biotechnology, now you're already studying with me. Furthermore, not every detail of replication is understood well. That's what I told you. In eukaryotes, the replication of DNA takes place at the S phase of the cell cycle. This is very, very important. Okay, a lot of PYQs come from this. That is in DNA, in eukaryotes. Okay, in eukaryotes, replication, okay, happens of DNA at S phase. S phase. Now, the replication of DNA and the cell division cycle should be highly coordinated. A failure in cell division after DNA replication results in polyploidy, a chromosomal anomaly. Okay, you will learn the detailed nature of origin processes and this in higher classes, and little bit you will also study in your principles of inheritance chapter. Okay, principles of inheritance chapter. Any doubts so far, students? Any doubts? Okay, now this is the diagram given here. Okay, it's the diagram given here. And this diagram is very important. See, look here. 5' to 3', you can see 3' to 5' we have formed the strand. Here you can see discontinuous strand. 3' to 5' you can see template DNA, that is a parental strand. Continuous synthesis, discontinuous synthesis, newly synthesis strands. This is your replication fork. So I have drawn a better diagram to show you about every enzyme that is going to work, but NCERT hasn't mentioned about all the enzymes, but it is important for you because again in biotechnology you will find it very difficult to understand if you do not understand this now. Okay, clear? So I hope replication was absolutely clear. Replication, that is semi-conservative replication, where your DNA, okay, semi-conservative replication where your DNA forms hybrid. Okay, DNA forms hybrid DNA, okay, where there is one parent and one daughter DNA. Okay, now how does this happen? This happens because of 4 enzymes mainly. Okay, 4 enzymes are going to be DNA DNA polymerase. You can write DNA dependent DNA polymerase. Okay, primer, DNA ligase. Okay, 1 second, students. I had to tell you about one more enzyme, SS, but I think boards it's not there. Okay, I'll just check your book. It is not there for sure. In biotechnology also they're not using those words. Now finding notes is a task. Prime, students, I've written primer, it's primase. Okay, spelling mistake I've written here also. Primase. Okay, topoisomerase I usually teach, SSBP I teach, but it's for NEET students. Okay, since you guys are both students, it's not required. Got it? Then helicase. I'll just tell you, okay, in case what if there is NEET students watching this channel. So students, topoisomerase is there and SSBP is there. That is single strand binding protein. Single strand binding protein. So this will actually avoid the strands to come back together, because see, we would have separated the strands like this. No, there is high chance that the strands will start coming back together, because you know that is one of the nature of your genetic material. Then topoisomerase will make sure that there is no supercoiling. Okay, there is no supercoiling, so this will avoid the supercoiling. Okay, got it? This will avoid the supercoiling. So I have just checked my notes as well, so that anything we miss I can just tell you guys. Single stranded. Okay, yeah, we call it re-annealing. Okay, here we call it as re-annealing, that is so that they do not come back to its position. Okay, re-annealing. Again, annealing, extension, all that you will study in your biotechnology, but this is different. Okay, re-annealing, where the strands will not come back together. Okay, they'll not come back together. And this one is very simple, right? They'll form the template. They'll form the new template. This is going to be the one who is going to lead the polymerase. Okay, lead the polymerase. Ligase is going to be the one who is going to glue. Helicase is going to be the one who unwind. Okay, unwind. So SSB understood, no? Re-annealing. Topoisomerase avoid supercoiling. Okay, avoid supercoiling, because DNA have the tendency, students, to come back like this. Okay, like best friends, no? They'll come back like this. So when they come back, problem will happen. Okay, let me check if this color is visible. I wrote nicely with the color. Ah, beautiful color, no? Okay, so that is the whole thing, whole concept. Now next is transcription. Okay, transcription. After transcription we can stop. So transcription. So what is transcription? That is, students, DNA became mRNA. Okay, and this process we call it as transcription. This process we call it as transcription. Right, now we are going to learn how transcription happens. See, the process of copying genetic material from one strand of the DNA into RNA is termed as transcription. Here also the principle of complementary governs the process of transcription as well, except the adenosine complements now forms the base pairs with uracil instead of thymine. However, unlike in the process of replication, so here what they're going to say, see, if I'm going to form an mRNA, simple thing is going to happen. Let's say this is your strand, this is your DNA strand. You have to form the mRNA because you're going to do transcription. Okay, what is going to happen? Here if you want to form mRNA, A will code with U, T will code C, U, A. Understood? U is your uracil. Understood. Thymine. So that's what the statement is. NCERT statements are little complex, but do not worry. So, however, unlike the process of replication, which once set in, the total DNA of an organism gets duplicated. In transcription, only a segment of DNA and only one strand is copied into RNA. I'll tell you about this, okay? Now, why both the strands are not copied during transcription has a simple answer. First, if both the strands act as a template, they would code for DNA molecule with different sequences. Remember, complementarity does not mean identical. And in turn, they will code for proteins. The sequence of amino acid in the proteins would be different. Hence, one segment of DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. Second, the two molecules, if produced continuously, would be complementary to each other, hence would form a double-stranded RNA. So, what they are trying to say, students, why? Okay, transcription I told you, ma'am, transcription I understood, that is DNA is going to become mRNA. But did I tell you how many strands of DNA? No, right? See, students, it goes like this. There is a rule in our body. The rule is that one DNA molecule, okay, one DNA molecule will code for one set of proteins. Okay, one set of proteins. Now, the question all of you will ask is, ma'am, why aren't we taking two strands? Let's say I take this strand. This is my template strand. Okay. Now, obviously, DNA has two strands now. So, this, let's say I take one more strand, which is going to be the coding strand. Now, students, if this strand forms an RNA, if this strand forms an RNA, okay, strand 1, and if this strands form an RNA, I'll draw it with a different color so there is no confusion. Okay, if this one forms another strand which is called RNA2, don't you think these two strands are complementary to each other? We just now studied semi-conservative. Same way, this is one strand of, let's say, 5' to 3'. I only made this, okay? Do not think that, ma'am, from where did you find this? Now, 3' to 5'. What is my strand going to be? My DNA is going to be T, C, T, A, G, C, T, A. This is my DNA, okay? This is my DNA strand. Now, let's say I used up this strand. Okay, I used this strand, that is 5' A, G, A, T, T, C, G, A, T, 3'. This is the DNA, okay? This is the template strand. So, this is going to be template. Now, if this template strand is going to form the mRNA, what is it going to be? U, C, U, A, G, C, U, A. Okay? It is going to be 3' to 5'. But this is an mRNA. Okay, this is an mRNA. Now, students, this is one strand I have taken as the template strand and form. Now, this will code for protein, one set of protein. Okay. Now, if I take this strand also, the second strand of DNA also, that is 3' to 5'. No? T, C, T, A, G, C, T, A. Okay. And I form the RNA 5'. See, it is going to be A, G, A, U, U, C, G, A, U. Right. 3'. Now, look at these strands. Okay, look at these strands, students. You will notice that this and this are the same. See, UC, TC, UAA, GC, TA. That is, only this uracil is replaced by thymine. So don't you think the mRNA that we are going to form is going to be complementary to our coding strand? So this is my mRNA, okay, and this is my coding strand. Coding strand means strand which is not going to help in the formation of mRNA. This is going to be coding. So these two are actually the same strand, same thing, just that U will be replaced by T and T will be replaced by U. correct? Now if I use both the strand, there is chances that this RNA and this RNA will bind with each other. They both will become complementary to each other. At the same time, this is going to code for one set of proteins. Okay, something like, you know, A, G, so like it's going to code for serine, valine and something. Okay, this fellow will code for another set of protein. That cannot happen, because the rule is from one molecule or from one strand of DNA I should get one set of proteins. So to follow this rule, we use only one strand that will form as the help us in transcription, that is called as the template strand. So coding strand and mRNA strand are exactly opposite to, I mean, same with each other, just that thymine is replaced by uracil. Okay, thymine is replaced by uracil. Is this clear? Now we'll go to transcription. Now students, transcription unit, okay, transcription unit, that is a transcription unit in DNA, okay, is defined primarily by three regions in the DNA. What are the three regions? Promoter, structural gene and terminator. Promoter means what? Someone who is promoting you. Okay, promoter is someone who is telling you do it or you have to do it. So it is kind of, promoter is the region where initiation is going to happen. So this is the unit. This is the unit and this is going to be the process. Yes or no? This is going to be the unit, this is going to be the process. So promoter means the one who is going to initiate, okay, initiate the reaction. So promoter region is where the initiation is going to happen. Promoter region is where initiation is going to happen. Okay, promoter region is where the initiation is going to happen. Then you have the structural gene region. Now students, you only tell me 2.2 meters of DNA. 2.2 meters of DNA. Do you think the entire DNA has to become mRNA? Entire DNA has to become mRNA? No, we just need a small portion of the DNA that is going to become mRNA. Like this portion is the portion that is going to become mRNA. That region is called the structural gene region, and that is where the elongation of DNA happens. Okay, elongation. So remember, this is the unit, this is the process. Unit is promoter, structural gene, terminator, and process is going to be initiation, elongation and termination. Clear or not, all of you? Yes? Okay, chalo. Now structural gene, after that we have terminator. Okay, terminator region, what is going to happen? Termination. Terminator region, what is going to happen? Termination is going to happen. Yes or no? So the unit of transcription, if they ask you, you will write a promoter, structural gene and terminator. Got it? Now let's see what is this. Okay, now students, look here. There is a convention in defining two strands of DNA in a structural gene of a transcriptional unit. Since two strands have opposite polarity, the DNA dependent RNA polymerase, because I have to form mRNA, I no more have to form DNA, so I am going to use RNA polymerase. This DNA dependent RNA polymerase also catalyzes polymerization in one direction, that is from 5' to 3'. The strand that has the polarity 3' to 5' will act as a template. Okay, so RNA polymerase also catalyzes the polymerization in only one direction, that is from 5' to 3'. Okay, the strand that has the polarity, okay, the strand that has the polarity 3' to 5' will act as a template and is also referred to as the template strand. The other strand which has polarity 5' to 3' and has the same sequence as RNA, I told you, except thymine in the place of uracil, is displaced during transcription. So strangely, this strand which does not code for anything is referred as coding strand. Okay, NCERT is making joke telling that you do not code for anything but your name is coding strand. So all the reference point while defining a transcription unit is made with coding strand. To explain the point, it is hypothetical sequence. So here you can see, students, 3' to 5' strand is going to be template, 5' to 3' strand is going to be coding. Okay, so look here. So this is the upstream. Okay, we tell upstream, that is from the 3' to 5' and this will be the downstream. So here upstream, here you can see, here is where the transcription is going to start. Okay, promoter is here, it is going to start, then it's going to go to the structural gene, then go to the template strand, and then then terminator. So here you can see the template strand, here you can see the coding strand. So RNA polymerase will work from what? Will work on a strand which is from 3' to 5', but it is going to form a mRNA which is from 5' to 3'. Understood? Which is going to form a DNA from 5' to 3'. Now let's see what is this whole thing. Okay. So much of best students. Wait, I'll tell you from the help of a diagram. See, initiation. Okay, initiation. That is the promoter region. Okay, promoter region, students. If I have to promote something, someone should be there, no, to tell me come inside. Imagine I'm going to a zoo. Okay, if I'm going to a zoo, there should be a watchman, no, who will tell me that, okay, ma'am, come inside. This is where you pay the bill. This is where you do it, take the tickets. So same way, when initiation is going to happen, there should be some person who is going to combine with RNA polymerase to help in initiation. So that person is going to be none other than your sigma factor. Who is it going to be, students? Sigma factor. But it's sigma factor. Now the sigma factor is going to be the one who is going to move through this region and reach up to the structural region. Okay, second step is going to be elongation, where the mRNA will be formed. Okay, where the mRNA will be formed. Got it? So by then the sigma factor will leave. And then this RNA polymerase is like the train which does not have brake, because this RNA polymerase will be key. It will keep on going. Okay, it will keep on going. It will not know when to stop. So to stop this, to terminate this, okay, to terminate this, we will have, that is RNA polymerase plus rho factor. Okay, rho factor will come into existence. This fellow will combine and tell that, wait, you have to stop now. This is where your entire process stops, so just stop. So this is what is transcription. Okay, it can be very simply done, but then there is a lot of complexity when you read it. So see, the promoter and the terminator flanks the structural gene in the transcription unit. The promoter is said to be located towards the 5' end, that is called the upstream of the structural gene. The reference is made with respect to the polarity of the coding strand. It is a DNA sequence that provides binding site for the RNA polymerase, who promoter, and it is the presence of a promoter in a transcription unit, also defined as the template and the coding strand. By switching its position with terminator, the definition of coding and template strand could be reversed. The terminator is located in the 3' end, which will be called the downstream. Okay, so go back here and see. This is the upstream for the coding strand. Yes or no? 5' end. Here is where the promoter is there. So this will be the upstream. That is going to be at the 5' end. And this is going to be the downstream, where it is there in the coding strand 3' end. Okay, 3' strand is going to be the downstream. Upstream means what? Student, this way, up. Downstream means going downward. So starting and ending. Now, the terminator is located in the 5' end downstream of the coding strand. It usually defines the end of the process of transcription. There are additional regulatory sequences that may be present further in the upstream or the downstream for helping this. Now, the DNA sequencing code for tRNA and rRNA molecule also define a gene. Now, here I will not go through this. Okay, I'll just tell you directly with the help of this diagram. See, now you understood transcription. I'm sure all my students understood transcription. Very simple. Initiation was there. For initiation to happen, we needed a promoter region. That promoter is present on the upstream of the coding strand, which is at the 5' end. Okay, that is where what is going to happen. Sigma factor is going to bind with RNA polymerase. Happily, they are going to move forward. They will reach the structural gene. In the structural gene, that is the portion of the gene that will going to form the mRNA. That portion will form a new strand. That's a job, no? So transcription, when mRNA will be formed. Then this RNA polymerase will keep moving and it does not know when to stop. So in the terminator region, there is going to be a rho factor which will bind with the RNA polymerase and it will stop or terminate the reaction. Now students, in eukaryotes, what happened? The mRNA that we form, or the gene that we take, okay, the gene or the mRNA we form, okay, is going to have a problem. That is, we are going to get a split gene. Okay, split gene. Split gene means what? There will be, see, at ultimate goal is you have to code for protein. Yes or no? Ultimate goal is you have to code for protein, and that is the whole goal. In between, if you go for something else, then that is not anyone's problem, right? Same way, here, the portion that you're seeing in green, Okay, the the portion that you're seeing in green is going to be the introns, which is actually going to help us with coding for the proteins. Then in the middle you can see a pink color structure which is called the exon. Okay, introns, which is not going to help us for coding. Okay, not going to help us with coding. Then we have exons, which is going to actually help us for coding for the protein. See, why am I forming mRNA to form protein? If mRNA only is not forming protein, then whom am I supposed to think about, right? So look at this. Introns are not going to code. Then why do I want it in mRNA? So I will do a technique called as splicing. Okay, I will do something called as RNA splicing, by which the introns will be removed from the mRNA. Can you see, students? Exons, introns are there. Introns will be removed with the help of RNA slicing, because introns are actually nonsense codons. The nonsense part of it. Why? Because they are not going to code. Okay, they're not going to code for the protein, so they're not helpful for me. So I will remove it with the help of splicing. After splicing and removing it, I will get a newly formed fresh mRNA, and to that mRNA I am going to add a tail and a head and tail. Okay, now I'm going to do the heading, we cannot tell, so we call it as capping. Why is this done? So that I understand the polarity, as well as no other new unnecessary things come and join with it. So the capping is going to be done with the help of methyl guanosine triphosphate. And the tailing is going to happen with poly-A tail. Tail is going to happen with poly-A tail, and that is because of polyadenylation. Okay, understood? So capping is going to happen with methyl guanosine triphosphate, and polyadenylation is going to happen because of poly-A tail. By this we will get a newly formed mRNA, and this is not usually present in prokaryotes, but this is usually seen in eukaryotes. I'll tell you also one more thing, one more terminology that you need to know is gene. A section of a gene is called a cistron. Okay, students, if you study this chapter with me, I am 100% sure you will be able to do amazingly good in your boards as well as in your exam. See, because I'm going into the details and I'm letting you understand that this is what this is. Okay, see, what is a cistron? A cistron word is given in your NCERT. It's actually a small section of your gene. Small section of your gene. Now, in prokaryotes, one gene can code for multiple proteins. One gene can code for multiple proteins. So we call this gene as polycistronic. Gene. But the same thing in your eukaryotes. One gene can only code for one protein, because that is the rule of our central dogma, rule of your molecular biology, rule of our entire thing, okay, of our evolutionary relationships. So one protein, this will be called as monocistronic gene. Now students, the gene which had both introns, exon can also be called as the RNA can also be called as hnRNA. Okay, hnRNA, which is heteronuclear RNA. That is someone who had both coding and non-coding sequence. Okay, coding and non-coding. So coding is the exon, non-coding is the intron. Okay, then splicing and tailing, you can remember. Capping, that whole thing, what we did is called as RNA processing, which you do not see in prokaryotes because they are not that complex. So RNA processing, you usually see it in eukaryotes because we have split gene. Okay, because of the presence of split gene. I hope this is clear. This is your transcription. Okay, this is your entire story of your transcription. So story, we will write the model of the story here. Okay, what does the story say? The story says that transcription has 3 units: promoter, structural gene, and terminator. Okay, now each of them, this is here is where initiation will happen, and this is where your sigma factor is there. Okay, this is where elongation will happen. Okay, here is where your structural gene, no need to write that. Okay, then here you have termination. Okay, termination where your rho factor is there. And who is going to do transcription? Who is going to do transcription? Transcription is going to be done by your RNA polymerase. Students, you can write DNA dependent RNA polymerase because it's going to work on a DNA, right? DNA say it is going to read and form the mRNA. RNA polymerase. Okay, this is the three things. Now rho factor will be there, done. Then here you have to remember about RNA processing, which you see, because RNA, we have a gene. Okay, we have a gene which is a mix of two things. Okay, mix of two things where you have exons, introns, exons, introns. So to remove these people, okay, to remove these people, let me do one thing and write it down. Okay, to remove these people, we will do a process called as splicing. Okay, a process called as splicing. This splicing here, exon, intron, exon, intron, exon. This will end up giving us a beautiful strand of mRNA that is only filled with exons. Then we will do capping, okay, and we'll do tail, okay, that is with poly A tail and your monoadenosine triphosphate. Okay, monoadenosine triphosphate. Then you can remember monocistronic, that is seen in eukaryotes, where you will form only code for one protein. Then we have polycistronic. Okay, this is seen in prokaryotes, which you code for many proteins. And RNA processing is only seen in eukaryotes. Clear, all of you? Okay, this is your entire transcription. Yes, so we have completed transcription. Okay, this I'm not reading because this is the same thing I told you. And yes, so that's it for today, students. We have completed transcription. Tomorrow we'll start with translation and we'll continue further. So thank you so much for joining. I hope the session was really helpful. Do not forget to like and share the session as well. It gives me a lot of happiness when you guys like the session and when you guys tell me how the session was. So do not fail to do that. See you all in next class. Thank you so much for joining. Bye bye.